our next speaker, Dr. Ken Kosik, is going to further look at a particular, talk about studies done with a particular group of individuals in Colombia. And Dr. Kosik is actually a physician scientist, and he completed his BA and master's degree actually in English literature at Case Western before going to medical school at the Medical College of Pennsylvania. And his early work was all done on this early familiar Alzheimer's disease. And what he has now been able to begin to do in a novel way with other group of individuals is to take a look at how can we try and use a very restricted population and perhaps learn from them about the mechanisms of the disease, but also target some treatments for them and begin to be able to see very readily whether these treatments are going to work or not. So he has also been co-founder of Learning and Brain Conference, and really has a long-standing interface or interest in neuroscience and education. So his accomplishments in the past have won him recognition internationally, and again, he has also been a recipient of the Metropolitan Life Foundation Medical Award, which is a very prestigious award. Also the Zenith Award and the Temple Award from the Alzheimer's Association. The Whitaker Health Sciences Award from MIT, and we could go on from that. He's also noted in Santa Barbara, where he's at right now, as a professor at the University of California, Santa Barbara. But in that city, he has gotten an Innovation Star Award, so congratulations for that as well. And a lot of his work has been picked up, and you might have seen some of this in other media, such as the New York Times, the Wall Street Journal, CNN, and those things. So now you get to have that straight off from the original innovator, Dr. Ken Kosik.
Well, thank you all. It's really a pleasure to be here, and I'm just thrilled to see so much interest in this topic. I want to tell you a story today that goes back, probably more than 30 years. About 20 or 25 of those years, it involved a extremely close collaboration between myself and Francisco Lopera, a neurologist from Colombia, where together he first discovered, and then we together characterized what is the largest family in the world with an inherited genetic form of Alzheimer's disease. You see here a picture of just one member, one member of this family. When I use the word family, I use that very loosely. This is a family of about 5,000 people who are scattered through a remote area of Colombia called Antioquia. And and we know they're all related genetically. They're fourth, fifth, sixth cousins with each other. And they have this, they get Alzheimer's disease at about age 45 or 50. And because the families are so large, it's not uncommon for us to see a couple, to get a history of a couple who might have met decades and decades ago, got married, had a large family, like many people. 12, 15 children is not uncommon. Little did they know when they first married that he carried this gene. After they've had their children and now he's getting into his 40s, he comes down with Alzheimer's disease. He may die, another 20 years go by, he may die. She was spared, and she is now a grandma. And her children, who are now getting into the age of risk, are coming down with Alzheimer's disease. It's not uncommon for us to go into a village and find a grandma taking care of 3 or 4 of her children with Alzheimer's disease. And it was these kinds of poignant stories that first triggered the interest of Pam Bellick at the New York Times to come down with us and do some work there and present this story to the internationally. And these are some of the, two of those frames are taken from that particular piece that was written by Pam. The other thing that I always find uncanny about whenever I'm there and looking at this family is the relationship. You've heard these phrases, art imitates life and life imitates art, and Gabriel Garcia Marquez, who actually Nobel laureate, recently died of Alzheimer's disease, wrote in his most famous book, 100 Years of Solitude, about a fictional town called Macondo, where the people would forget. They forgot things over time, and that's in the opening chapter, and it's, there are these uncanny relationships. He knew nothing about this family, of course, but these kinds of relationships between fiction and real life are always intriguing. So here's a picture just of a small piece of the family tree. If we drew out the whole family tree, it would fill the walls of this room easily. This is an autosomal dominant trait. That is, if you have a parent that has it, the offspring have a 50% chance of getting it. They're drawn as separate families, but we've now been able, because of a lot of the work we've done in genomics and better and better family trees, we actually can link almost all the families together. A number of years ago, in collaboration with Alison Goat, we found the mutation that all the members of this family carry. There it is in presenilin. It changes a glutamic acid at position 280 to an alanine. And that, we refer to that mutation as a private mutation because this is the only family in the world that has that mutation. I'll tell you a little bit about the origin of that mutation in a moment. But it is in presenilin, which carries many other mutations at different sites, all leading to familial Alzheimer's disease. Now, just so that, just to refresh you on your geography, this family is from Colombia. They're in the northern part of Colombia, up where you begin to see, up in that coastal region up to Antioquia. There's an isthmus there, that's Panama coming off from Colombia. And the capital of that particular state in Colombia is called Medellin, which you've heard of. About half of the family members have migrated in from the campo, from the countryside into Medellin. The other half are still out in the rather remote areas. So what we're going to do for the next few moments is take a little tour of how we go about collecting these family trees and samples and learning about the families. This particular meeting, this took place years ago. This is early on when I first started to go there, where we called some of the families together that live in Medellin. And here we are explaining to them the story of Alzheimer's disease and a little bit about genetics. And the reason I like to show this slide is because you see there a woman in a wheelchair who has the disease, carries the mutation, and behind her is her granddaughter, who's still very much involved in her care. I am continually struck in Colombia how warehousing of elders, and particularly elders with dementia, does not occur. There are no nursing homes, there are no assisted livings, there are certain assisted living facilities, certainly none out in the countryside. And children see elders right up until the time they die. They are right in the home, and there's no walling off. And you can see that in the way, sort of the tenderness of the granddaughter. The families that are from Medellin live in these hillside shanties around the town. Medellin is actually a beautiful city. It's in this gorgeous valley, very green. But on the hillsides, just like the favelas in Brazil, there are these shanties where most of the families live. And you can see what it looks like. They're built in this very steep landscape. And when we would go out there to visit, you can see in the first of those shots there is the back of Lucia Madrigal, our nurse, going down into one of these little alleys and about to enter through that door. And inside that door is lying in bed this woman who has advanced Alzheimer's. She's now vegetative. Her daughter is there taking care of her. Her daughter is 17 years old. She's been taking care of her mom for now 9 years. She, unlike this particular family, the families are huge, but this family's not. This young girl has been taking care of her mom almost solo for almost a decade, right up to this stage. From Medellin, we will often travel out to the countryside, and one of the local provincial capitals is called Yarumal, which is about a 5 or 6 hour drive from Medellin on these winding roads through the mountainous countryside. And here's the town square of Yarumal. Here we are again calling the families together in a local hospital to talk to them about the disease. They have a lot of their own folk legends about this disease. They sometimes think that they get the disease because they might accidentally touch a tree, but they don't know which tree it is. Or even worse, they sometimes think that they will stigmatize individuals with the disease because they think they can catch it by touching someone with the disease. So we try to tell them a little bit about genetics and how this all works. And when we did start to talk about genetics and how this is a dominant gene, so you have a 50-50 chance of getting it, just like a woman has a 50-50 chance of having a boy or a girl. The gentleman you see there in the first row, he stood up and he said, I have a comment. It's not a question, I have a comment. I want you to know that in my family, we have 12 children. And in our family, only 3 of them ended up getting Alzheimer's disease. So I want to thank you, the families, he pointed to the other side of the room, who also have 12 children and have 7 or 8 affected. This is a really interesting idea about gratitude and where the sources of chance arise. Before Gauss discovered the curve, the normal curve, the bell-shaped curve, people thought that the beauty of that curve, we knew that if you flip a coin, it was still 50-50. But where does the bell-shaped curve arise? And he wanted to give a gratitude for what is really, now we understand, is a matter of chance. He also asked, he said, I also have a question. And this was a tough question. He said, I want to ask you something else. Is it okay if we marry our cousins? And, I said, well, I have a little bit of a genetic orientation, and I think it's not such a good idea. And then he said something that I could not respond to. He said, well, we have no one else to marry. But many of them live in small towns, in small little villages, and they don't travel very much, and there is an enormous amount of consanguinity. Now, just remember, though, this is a dominant mutation. So when we worry about consanguinity, we're worried about recessive mutations now coming on and causing pathology, and they certainly have their share of recessive mutations, but in this case, that's not the issue. However, I'm going to tell you in a few more slides a little bit about some homozygotes we discovered. Anyhow, here is another family. Just to show you how remote it gets. This family, here we are gathered together here in Sopetran. In the middle is Lucia again. But this family is not from Sopetran. They're actually from Moron. They came in by horseback over those mountains that you can see in the background because there's no roads. And we began to get information from them there in this area. Here's another interesting story. We have a social program down there because there's a lot of poverty. Most of these people are extremely poor. And there was a woman who lived out in Betania which in the bottom, you see that green picture. You have to travel actually the opposite direction from Yarumal, another 5 hours, way out in the countryside to see a woman with Alzheimer's who recently died, and she had been given a bed as a hospital bed from our social program. And when she died, we went out to collect the bed. And we drove out there, and we got back to Medellin pretty late at night and came in to try to deliver the bed to another family who lived in the outskirts of Medellin in a little area called Manrique, which you see in the upper pictures there. Well, the delivery of the bed at about 10 o'clock at night in Manrique was a very public event. This was one of the biggest things going on. You can see the people all gathered outside there from the street. There's people coming inside. None of those people are related to the woman who's going to lie on that bed. This is, the delivery of the bed was very public. Things like HIPAA and privacy in these places does not exist at all. Everybody knows what everybody else is doing, and delivering a bed is a very interesting event that everybody will attend. So and it's another facet of doing things in Colombia. Here we are going out to a town. It's a place I always wanted to go. They never let me go there until recently. It's a small town called Angostura. This town's had a little bit of trouble, but there are a lot of people that come from this town. This is where, in this town, here's a picture from the town. There's Lucia drawing blood and a woman getting a neuropsych test. And in this town is where, in one of the churches, is where Padre Marianito lies. He is considered to be on his way to sainthood. And the reason I'm showing this is not because really it's with regard to Padre Marianito, and even more so, there's other people in all of the villages who are the so-called curanderos. Actually, they call them witches, actually, but they're not witches like we think. This is the bruja, and they go around. They can be good witches or bad witches, and they will, and they're very important for caring for or looking to the welfare of individuals. So you have to hedge your bets if you're in Colombia. You have to turn to traditional medicine, but you also have to deal with the healers as well, because they don't know what's true. So, we may be saying that, yes, you should take this medicine, and this is the way Western medicine works, and then the healer says, yes, but you've got to do this and that. And unless you sort of work together, nothing's going to work. You can't disparage their belief system. So it really is a matter of collaborating with a group of people in medicine that we're not used to collaborating with. Here we are again in calling together people in Angostura for, again, the same type of presentations, but now we're getting closer to the present, and this is what you heard about during the introduction, where the group at Genentech and Banner had gotten in touch with me to implement a clinical trial down there on this family. It's the largest family in the world, and they get their disease at a very set time. So there was an interest in a clinical trial, and in this picture you can see now we've invited over on the right-hand side of the screen Pierre Tario from Banner to help us in doing a clinical trial in this group, which has now been launched, and I'll tell you more about that in a few moments. I want to say show you one more picture from the tour we're taking. Right here. Here we are out, there's Dr. Lopera in the center, and this is a children's group that Clara Monica, Dr. Lopera's wife, formed. These are all the children that come from the families. We have two children's groups, one are more teenage, like this group, and one that are even younger. Every single one of these kids you see here has got 50% chance of getting the disease. And now they know each other a little bit, and they have this sort of support group. Okay, let's get to some of the statistics and the genetics. And work done by Natalia Castabuena. We know that the onset of this disease in this population due to a mutation in presenilin occurs like clockwork. It just, they get MCI at around 45, they convert to Alzheimer's at around age 49, and they also have these earlier symptoms that we've begun to characterize as pre-MCI. And then, in collaboration with Adam Fleischer, he was able to take a set of these people and bring them up to Banner in Arizona and do PET imaging on them. So here are some of the pictures of something that you already know. There's nothing astounding yet on this slide that's coming up on the next slide. This slide is really just showing you that all that yellow and red indicates that people with dementia on the top or MCI on the bottom row already have a head full of dementia. I mean of amyloid, and probably causing dementia in some way. But not definitely causing dementia, because MCI, mild cognitive impairment, also, as you see there, has an awful lot of amyloid in the brain. From the time you get MCI onto Alzheimer's disease, the amount of amyloid is already on a plateau. So there's other things going on that are leading to the dementia, probably not just the amyloid. The reason I wanted to show this, though, is really on the next slide, right here, because here you can see that people as early as in their early 30s, and maybe late 20s, but certainly early 30s, and definitely 35 to 39, are already developing amyloid in the brain. That is long before, at least a decade, if not more, a decade before they have any symptoms of dementia whatsoever or memory loss. These individuals are already beginning to deposit amyloid in the brain. So this disease begins long before there are clinical symptoms. So findings like this, that is the presence of amyloid early on, the fact that they get the disease like clockwork and will inevitably get it if they have a mutation, has all led to the idea that doing a clinical trial here would be a good idea. And this coincided with the phase 3 failure of many drugs that have been tried by pharma, leading to a paradigm shift in the entire field that what we really should try to start to do is to prevent Alzheimer's rather than to cure it once somebody already has it. We may require a different treatment to treat Alzheimer's when a person already has it, because they've already lost a lot of brain cells and it's a much bigger challenge. But maybe we can do better if we can somehow implement therapy before the disease strikes. But who do we treat? Well, in Colombia, we know who to treat. We know who has a mutation, and we know when they're going to get it. Even if we knew that somebody was going to get the disease in a sporadic population, we just don't know when they're going to get it. If we look at elders that are 75 years or older, they have a chance of getting it, but we don't know when they're going to get it. Here we know that. So this clinical trial was maybe 2 years ago, was funded, and the drug that's being used is an amyloid antibody called crenezumab, made by Genentech under the direction of Carol Ho, who is the lead investigator in Genentech, in collaboration with the group at Banner led by Eric Ryman. And here is one of the individuals in this family. She's given permission for this picture. She's sort of been the poster child for this project. There's a lot of pictures of her in the media. And here she is getting her Amyvid scan when she was in Arizona. So the trial has begun, and it's going to run for 5 years. We're not going to know the answer for a little while yet, but to get into the trial, you cannot have any symptoms. You just have to be completely pre-symptomatic, and there's about 300 people enrolled. There are 100 people that have the mutation and will get drug. There are 100 people that have the mutation and will get placebo, and there are 100 people that have no mutation and will get placebo. That way, they don't know who has the mutation and who does not. This has been a real challenge for us throughout all the years I've been working on this. That is, do we inform people about this mutation? To inform someone about this kind of an issue is really a very, very serious matter. I remember when we first discovered the gene, I was in one of the homes of one of the families, and I was talking to a kid about 24 years old, and I said to him, you know, we've recently found the gene. You're from one of the families. Would you want to know or not? He said, yeah, I want to know. So why do you want to know? And he didn't answer me. But then he went into the kitchen, and Lucia was there, and he said to her, you know, if I had that gene, and he put his hand to his head like I said, he'd shoot himself. He's 24 years old. He has another 24 years until he even gets his first symptoms. Genetic information is fire. And we have to be very careful about telling individuals in Colombia, because there are no genetic counselors there. We hope there will be, and we would like to get them as part of this program, but at the moment, if they want to participate, they've had to agree that they would not be informed about their genetic status. That may strike you, and it strikes me, as somewhat paternalistic, but we don't have a good answer for this yet. And that has been brought home to me many, many times, when more than once a woman, 28, 31 years old, has come up to me crying, saying, I want to have children, but I'm afraid. So we have to resolve this, and it's certainly a very present matter that's still going, still in play. Okay. I had said before that this is a private mutation. It's just in one family. They all have this mutation. Well, where did this mutation come from? So we've now sequenced about 130 genomes of carriers of this mutation. And what you see here is what is called the line in the middle there, where there's that's empty space, all those lines represent different individuals. The different colors, the stacks represent different individuals, and the piece right in the middle is different individuals and a stretch of DNA from each of them. And that stretch of DNA comes right from the place in the chromosome where the mutation lies. So what we did was we took the DNA segment from all of these individuals in that stack and lined up one stretch of DNA right around the mutation. And you can see where that space is in the middle. They all have the mutation, so it was all the same there. But then you can ask the question, how far out from the mutation does their DNA sequence remain identical? That's what IBD stands for, identity by descent. That is, and it turns out that all the people in that stack have a very long stretch of DNA around the mutation that's exactly the same. Of course, for some it's short at the top, for some it's very long, but even the short pieces are pretty long for having your DNA being exactly, nucleotide for nucleotide, the same as the next person down in that stack. What does that mean? That means that there was a single founder for this entire kindred, because not only, it wasn't that this mutation arose independently in different people, but they all inherited that same stretch of DNA in every single one of these people. So from this data, we know that there was a single founder, a founder effect. We can also, the reason those bars along there are different lengths is due to recombination, DNA exchanging. And recombination occurs at a certain rate. So we can also begin to calculate through a number of statistical techniques, sort of what are called coalescence techniques, how far back that mutation goes, when that a mutation first arose. And when we do that, it goes back about 18 generations, maybe to around 1,500. So so we know there was a single individual around 1,500 that gave rise to this family. So let's look at what's happening around that time. This is the population structure in Antioquia and all of Colombia. Is what's called an admixture. There's the Amerindian population, and then beginning in 1492, Spanish invaders came. They were all males, and they had offspring with the Amerindian women, and several generations of influx of Spanish invaders went through those different arrows you see there from generation to generation and created an admixed population. We know that very, very clearly from the work of Andres Ruiz, whose name is at the bottom there, that in the Colombian population, on the mitochondrial genome, which has a maternal inheritance, you see Amerindian markers, and in the Y chromosome, which comes on the paternal line, you see the Iberian Peninsula markers, Spanish markers. So the history is absolutely clear from the genetics, absolutely. So the next thing we can do is to look at the markers around the mutation. Did this mutation come from the Amerindians or did it come from the Spanish? And you can do this local ancestry analysis, and when you do that, by comparing markers around the mutation from different parts of the world, the bottom line here is that those pink bars that you see that are very high, especially in the lower panel, they represent markers around the mutation that are from the Spanish population. It's very clear that the mutation came from Spain. So we know that, so we now know when and where it came from. And so when the Spanish came over, in missions, even Christopher Columbus touched around this area, but later missions, this is where they actually entered deep into Colombia. What I'm showing you here is Darien Bay. This has got to be some of the roughest area in the world. This is an area where the Pan American Highway ends down in the middle of Panama and doesn't pick up again until you're in South America. It is some of the densest, impenetrable jungle that exists. No roads, nothing. And that's the area where the Spanish invaders came in. It's somewhere, it's one of these individuals, one of these guys, who brought that gene, and then what they did was absolutely incredible. This is the roots, the different routes that the various Spanish invaders took as they traveled through Colombia, setting up settlements. And the first city founded in Antioquia was called Santa Fe de Antioquia. That is somewhere, I can't see it as well, but it's somewhere on that map. And it was founded 49 years after Christopher Columbus first landed. So in a mere 49 years, the Spanish invaders came across the ocean, landed in some of what even today is some of the densest jungle in the world, penetrated deep into a mountainous region full of ravines in Colombia, and founded a settlement which then gave rise to the people that are today what we call Paisa. Paisa are Antioquians, that's what they call themselves. And there were other settlements found by other conquistadors. So they all founded these different settlements in a very short period of time. It was just remarkable what was going on at that time. So as we're thinking, so let me just, so I'm going to come back to this idea about these settlers in a moment, but I want to tell you one more thing that we discovered in the genetics here that I had alluded to earlier. Because in going through these families, while we're on this topic of the genetics and the origins, we actually came across some families like this, where both parents had the mutation. And we had looked for homozygotes for a long time, and we thought that homozygosity must be lethal because we never found any, even though there was all this consanguinity. But then the first one showed up, right with that arrow there. And then 6 more showed up. So we now have 6 homozygotes, and so here they are. Most of them are young. The one back here, this is this one here is an 11-year-old girl, in which we got special medical permission to be able to test her DNA. But some of them, 2 of them are already older, around 35 to 40. They have dementia. There are 3 that are in the range, that age range, 27 to 38. They don't yet have dementia. They will inevitably get it. And then this younger one that has mild mental retardation. Now, this is a statistically meaningless statement, but I'm going to make it anyhow because we know that the onset of the disease in this population is so tight around age 45 or 50. And 2 individuals with homozygosity have an earlier onset that, as far as I can tell, homozygosity is compatible with life, but it may be pushing the onset a little bit earlier. That's about, I think, the most we can say. Maybe we can say one other thing, which is that of the 6 individuals, 5 are females. The probability of getting 5 females out of 6 independent births is like 0.09. So there may be, if males have homozygosity, there's fetal wastage. We don't know, but there is a little bit of a tendency here. So let me continue in this vein with the genetics and talk further about these people that may have a shift in their age of onset. The few people who may not get the disease at age 45 or 50, but get it later or earlier. Those what we call outliers. So we'll talk about using the genomics to identify outliers. That is, you heard from Jerry about whole genome sequencing. We did whole genome sequencing on an outlier population and other controls to see if we can find the gene that was associated with outlier status. And here's this bell-shaped curve where you might expect some outliers. This is the actual curve, the way it looks, where you have many of the patients in that white area with the mean age of onset and a few that are outliers in the green or the pink area. This is not exactly a bell-shaped curve. There's a little bit of a skew toward alleles that are protective. However, because the mean age of onset is so tightly clustered around 45 or 50, we think that any variant that will shift the age of onset is likely to be a strong allele, and that makes it worth looking for. So, and the heritability of age of onset is fairly high. So we're doing that right now. We have actually some loci that are starting to look interesting. You heard Jerry say about this amazing p-value of 5 times 10 to the minus 8th, which we think we're getting close to. But we still have a lot more work to do. However, it's a small sample. So is it going to be meaningful? Well, it may be, and here's the reasons why. The problem with many of the association studies are seen right here. There are several problems, but this is a big one. That is, you take these populations, diverse populations, like say everybody in this room, we all have different ethnic origins and different backgrounds, and if you have some variant in your genome, maybe it's harmful to you, but somebody else may have the same variant and it's not harmful to them. That happens all the time. So cases and controls can be sampled disproportionately, and therefore it's very hard to get an association when you have a very mixed population, because we all have different genes, and those genes may have different effects in each of us. It can be equally hard for the same reason, although it's a little tighter, to get statistical associations with a gene variant, even in an admixture population, for the same reasons. But remember, in this case, it's all one family. These are people who are all related to each other. So if there's a variant that seems to be doing something in one individual, it's very likely to be doing the same thing in the other individual because they come from fairly similar genetic backgrounds. And that's why we think we may be able to get some mileage out of these whole genome samples, even on a population that is considerably smaller than the ones that you heard Jerry talking about a moment ago. The other thing that we have going is you've heard about this technique now where you can, this was actually this technique received a Nobel Prize this past year. This is to be able to make stem cells from a skin biopsy. You don't have to take embryonic stem cells anymore. You can just take a skin biopsy from any of us and then convert your cells right into stem cells. And then we can make those cells into neurons, differentiate them right in the dish. And we've done that from some of the people that have different ages of onset in this population. And then we can actually begin to see by sequencing now which genes are expressed in those neurons. You know, we all have about 25,000 genes, but they're not all expressed in every single tissue. So we take a set that are expressed in these brain cells in the dish, and we see which genes might be at a higher or lower level. And when we look at this kind of analysis, and this is just, and we compare the clustering of the expression of all these genes, there's a lot of sort of statistical techniques behind this. This particular graph is called a principal component analysis. But basically, those blue circles there show you that there is a tendency for the presenilin 1 cases, that is these people that are carriers of this mutation, to cluster in their gene expression patterns differently than the controls. And so what we're looking at there basically are two blue circles done in two different ways. And the fact that the blue circles are different indicates that the gene expression patterns are clustering in a somewhat different manner. It's another approach in to find where gene regulation may be a problem. You heard that in the genome, we find these genetic variants someplace, but they could lie anywhere. We don't know what they're doing. That's why Jerry used the word causal variants. Suddenly his list got really reduced when he tried to find genes that were variant and also causing some sort of a phenotype. So by looking at these what are called expression QTLs, that's just a technical word, looking at the expression level of genes, we may be able to get more mileage out of figuring out what the variants are doing. So that's all a bit technical. Let me conclude by showing you one more topic that is intriguing me a lot lately, because this family with Alzheimer's disease, with regard to genetics in this area, is actually just the tip of the iceberg. What you see here is a disease map of Antioquia, and the red areas are where the families live with the Alzheimer's that we talked about. But there are other colors on that map. And those are other families that also have dominant mutations leading to serious neurological diseases. For instance, there's another family in a little town just outside of Medellin called Copacabana, which has also Alzheimer's disease, but a different mutation in presenilin 1. And this family is almost as big as the one that has gotten all this attention. And then we have other families that have other diseases. There's a disease called CADASIL, in which it's rare, in which individuals get strokes at a very early age. And it's known to exist that there are mutations in Notch3, a gene called Notch3. It's rare. There are two different Notch3 mutations among those colored populations there in Antioquia. There are a number of dominant mutations. Now that's pretty incredible. When you have an inbred population, as we said a few moments ago, you certainly expect many more recessive mutations because, of course, people that may carry a harmless mutation have children with somebody else that has the same harmless mutation, but having two copies of it now leads to a problem. These are dominant mutations. It means that all you need is one copy, one parent to have it. These mutations are not coming from consanguinity. Where are they coming from? Why are there so many dominant mutations in this population? Antioquia has about 8 million people. There's Amerindians and Spanish invaders. Where did all these mutations come from? So I don't know the answer, but what I've begun to think about is what is the comment on that slide there. That is, you saw that map where the Spanish invaders came into Antioquia, and they settled in all these different towns deep into Antioquia in these very in these areas that are tough to get to. They settled and they stayed there. That, in genetics, that's called a bottleneck. That means a small group of people gave rise to the whole population of descendants. When populations arise from bottlenecks, we begin to see mutations, even dominant mutations, emerging in these populations. We all carry serious mutations. We're just lucky we don't see it all. But in these populations that are all with a common founder, we may end up having an excess of dominant mutations. And that's something that we're very interested in studying further. So, to conclude, I'd like to go back to Gabriel Garcia Marquez and his 100 Years of Solitude, because you remember in this story, the people in Macondo lost their memory. But at the end of the chapter, there's a gypsy who arrives, and he brings with him una sustancia de color apacible. That means a substance of a gentle color, and he cures their memory problem. Well, I'm optimistic that this clinical trial will perhaps be the gypsy for these people. But if it isn't, I think we've still done a lot because we have the infrastructure now to do additional trials. And with that, I want to thank an enormous number of people here. There's too many people to thank individually. There's a lot of the sequencing was done with the group at Institute for Systems Biology in Seattle. The Banner group with the clinical trial have just been amazing. Genentech has also been just critically important, and my lab up in the corner there with Matthew Lally, who did most of the work that, the genetic work I showed you. The very large group in Colombia that's actually made this work, but by far and away, there's one person who just deserves so much of the credit here, and that's Francisco Lopera, whose name is up there in bold. Here are the people involved. You see there's basic labs there. Up in the right, there's Dr. Lopera in that picture, and in the lower picture with all the kids. But as I say, it's really Francisco who has been the soul and spirit behind all of this. Thank you very much.